All right, so the last category of microcytic anemias that we're going to talk about are thalassemias. And thalassemia is a problem with globins. So these conditions result in diminished or absent globin chain synthesis. My pen is being weird. Okay, globin chain synthesis. Everything up until now has been a problem with heme. And it has all involved issues with iron, in case you haven't noticed. So not enough iron, iron not getting into the red cells for different reasons, or too much iron. So now we're talking a problem with globin. And if you remember the normal hemoglobins, you know that the main adult hemoglobin, hemoglobin A, is made up of 2 alpha globin chains and 2 beta globin chains. And if you can't make one of those, you're going to have problems. So you're going to have not enough hemoglobin, so you're going to have a microcytic cell. And this has nothing to do with iron. It's not linked to iron problems at all. So thalassemia can have very ugly red cells, and you can see that in the picture. Very distinctive, abnormal shapes going on. So there are 2 different kinds of thalassemia. There's alpha thalassemia, and then there is beta thalassemia. And beta globin genes are found on chromosome 11, and there is 1 gene per chromosome. Oops. And alpha globin genes are found on chromosome 16, and there are 2 per chromosome. So there are a total of 4 alpha globin genes, and there are only a total of 2 beta globin genes. So you can see, I knew that picture was on here. I wasn't sure where it was at. Alright, so then we have a chart of the various types of thalassemias. So because there are 4 alpha globin genes, you could be missing one of those, in which case you would be called a silent carrier and you probably wouldn't even know that you have it. Or you could be missing 2 alpha globin genes, or you could be missing 3 alpha globin genes, and then that's called hemoglobin H disease. Or you could be missing 4 alpha globin genes, and that's called Bart's hydrops fetalis, or hemoglobin Bart's, which is incompatible with life. You have to have at least 1 functioning alpha globin gene to be alive. So in beta, you can have just 1 defective gene, and that is called beta thalassemia minor, or you can have 2 defective genes, and then that's called beta thalassemia major. So the reason why is for the alpha globins, there are 4 genes, and for beta thalassemia, there are 2. So that's why there's 2 different categories versus 4 different categories. So like we said, it's a microcytic process. They'll have a microcytic form of anemia. They have normal iron status. It's not related to iron problems whatsoever. It can affect multiple organs, and we'll talk about that when we get to the different categories of thalassemia. And thalassemia can require patients to have a transfusion. And they might end up with problems with not having enough normal, or we could say functional, hemoglobin A being produced. We know that all the normal adult hemoglobins have alpha globins, and then hemoglobin A has beta globins and alpha globins actually. Alright, so that's kind of an introduction to that. I keep trying to slide. So the alpha thalassemias are hereditary, and they are found in certain populations. And there is a high incidence in the Asian, Saudi Arabian, and Filipino populations. The alpha chain is responsible for building adult hemoglobin. So normal adults have 3 kinds of hemoglobin. We have hemoglobin A that has the 2 alpha and 2 beta globins, And then we have hemoglobin A2 that has 2 alpha globin chains and then 2 delta globin chains. And we have hemoglobin F that has 2 alpha globin chains and 2 gamma globin chains. So each one of these adult hemoglobins have alpha globin. So if you don't have any alpha or very little alpha, then you're going to be in big trouble. So the worst one of all that is incompatible with life is Bart's hydrops fetalis. And they have no alpha globin chains. And so this is incompatible with life. If a woman would get pregnant and the fetus would have Bart's hydrops fetalis, the pregnancy will end in spontaneous abortion, also known as miscarriage, or the baby will be stillborn. The next category, where they have one functional gene, is called hemoglobin H disease. So here you're only born with one functional alpha chain. And you've got 3 genes that don't work. And you might have a little bit of hemoglobin A, but most of your hemoglobin is going to be hemoglobin H. So because you have an excess of beta chains and no alpha chains, and those beta chains all stick together to one another and they fuse, and then you get hemoglobin H. So it's non-functional and it can't carry oxygen. So hemoglobin H makes clumps in your red cells. And it says on the little section here under hemoglobin H disease that hemoglobin H inclusions make the red cell look like pitted golf balls. You know how golf balls have those little divots in them? That's what their red cells will look like in a patient who has this. And that's a good thing to remember. And these people have a severely low hemoglobin, and they will definitely have microcytosis, and they are trying to make blood, so they will have an increased reticulocyte count. And because of this bad hemoglobin, the spleen gets really involved in trying to get those hemoglobin H particles out of the red cells. So they'll have splenomegaly along with that too. All right, and these last two here, alpha thalassemia trait and then the silent carrier, they're not as bad. They're not so bad. In the silent carrier, you have 3 functional globin chains and are considered hematologically normal. Even if you have only half of the alpha globin genes, you will probably just have some very mild anemia. And when you're born, you might have an increased amount of hemoglobin Bart's, which is 4 gamma chains. And after you start maturing and you don't have that large amount of hemoglobin F anymore, then you won't have that hemoglobin Bart's present. So up at the top of our depiction here, you have a mom and a dad. Mom is the yellow, dad is the red. Mom has alpha thalassemia because she's missing 2 genes. Dad is a silent carrier because he's missing only 1 gene. Next is what can happen if they have children. So they could have a normal child. The mom gives a chromosome that has 2 functional genes, and the dad gives a chromosome that has 2 functional genes, and so they would have normal hemoglobin. They could also have a child with alpha thalassemia trait, just like what the mom has. 2 of the genes are missing. They could have a child that is a silent carrier, just like the dad is. Or they could have a child with hemoglobin H disease, and in this case, then there would only be 1 functional alpha globin gene. All right, next we're going to talk about beta thalassemia, and beta thalassemia major is up first. So just like alpha thalassemia involves alpha genes, beta thalassemia involves the beta genes. So other names for beta thalassemia major are Mediterranean anemia and then Cooley's anemia. And there's little or no beta chain synthesis. they can't make hemoglobin A. So what happens is hemoglobin A2 and hemoglobin F have to increase to take its place. So the majority of the hemoglobin for these patients is going to be hemoglobin F. And over on the right hand side you can see a picture of a skull. And a person with beta thalassemia major, their body will try to make more blood, and it knows that they're anemic, but what ends up happening is that all of their bones end up expanding. So this skull shows that expansion. It's much thicker than it should be. And in your book it says that they might have some unusual facial features as well. And they can have a long mandible and then a big forehead too. And then in the lab, we will see microcytic and hypochromic red cells. They'll have polychromasia because their bone marrow is going to be putting out more cells. Nucleated red blood cells might even be present. They will have Howell-Jolly bodies, and a lot of that is due to things then going on with their spleen. And their hemoglobin is going to be between 6 and 9 milligrams per deciliter typically. And babies that have beta thalassemia major often have failure to thrive. And then patients, enlarged spleen, anemia, and jaundice. So what can be done for these people? Well, they can try to transfuse them, but it might lead to iron overload problems. And if this happens, then they will have to have iron chelation done. And they're working with bone marrow and stem cell transplants for patients that have this condition. Then beta thalassemia intermedia is the next one. They're not as severe, but they do develop problems maybe later in life. They may have a larger spleen and mild bone changes. And then we have beta thalassemia minor, also known as beta thalassemia trait. And this is where you only inherit one abnormal beta gene, and that is what heterozygous means. And a lot of these people look like they have iron deficiency, except that they are going to have this high red blood cell count. And also their iron stores are going to be normal, and so that's because it doesn't have anything to do with iron, so that's why that's not affected. So when they say that it mimics iron deficiency anemia, initially it looks like it because of the symptoms and the fact that they have microcytic red cells, but then when you get to checking those actual iron results and looking at their red blood cell count too, then that can help tell you that that's not what's going on. They have a high red count, and you wouldn't associate that with iron deficiency anemia. They will have a decreased hemoglobin and hematocrit, and they'll have an increased amount of hemoglobin A2. It's not an iron problem, so giving them iron is not going to be of any benefit to them. And you might see some basophilic stippling or target cells under the microscope with these patients. So this would be what beta thalassemia trait can look like. Mom on the left has beta thalassemia trait, as does dad on the right. They are each missing one gene. They could have a child with normal hemoglobin, they could have a child with beta thalassemia trait that looks just like they do, or they could have this beta thalassemia major where there are no functional beta globin genes. And in your book it talks about this beta 0 where no chains are produced, and then beta plus where only a little bit of chains are produced. So there are some other little odd side things associated with beta thalassemia that people can have. Alright, so lastly we have an overview of our microcytic disorders. So you can see the separate disorders listed there as their abbreviations. And so AOI, anemia of inflammation, or ACD, anemia of chronic disease. Hereditary hemochromatosis is HH. IDA is iron deficiency anemia. SA would be sideroblastic anemia, and then thalassemia down at the bottom. So iron tests are very useful with the microcytic anemias. The doctor will do these tests if a patient comes in with anemia and they have a low MCV. So they do the CBC first, and then we'll move on from there. And I know I've talked before about reflex testing, where we order tests based on tests that are completed. previously. So for example, if a patient has a rapid strep ordered and the test is negative, this should lead to a reflex test for a throat culture to catch any false negatives. Same thing with urinalysis. Something shows up abnormal with there, then we're going to do the microscopic exam or so on. So if a patient has anemia and they have a low MCV, then the doctor is going to most likely order iron tests. That would be the kind of reflex test for that. And other key things that you do need to know, those physiologic features, things that I said were really important to remember about each one of these. And then those definitions, clonoikia, that's the spooned fingernails that are associated with iron deficiency anemia. Chelosis, that's the having to do with their mouth. Also iron deficiency anemia. And then lastly, we have pica, where you want to eat things that are not food. And that brings us to the end of chapter 5.